logic gates. They are obligate intracellular parasites, which is just a fancy way of saying they are dead code until they plug into a compatible port.
Dead code is such a striking image. Because usually when we talk about viruses, we're talking about the chaos they cause, the fever, the pandemic, the days off work.
The symptoms, yeah.
But the mission for this deep dive is to ignore the symptoms for a moment and look at the cause. We want to decode the logic of the hardware. How does the physical structure of a virus dictate its behavior? How does it hack the cell? And the part I found most unsettling in the reading, how do they hide in the architecture of our own nervous system?
That neurovirology section is where the stakes get very, very high. But you're right, you can't understand how they hide in the brain until you understand what they are made of.
So starting at square one.
If we start with the definition, a virus is quite literally a package of genetic material wrapped in a protein
You
coat.
can do
That's
it.
There's no
Let's
metabolism.
work today
This is
to
the
make
part I want to stick on
tomorrow
for
a
a second.
better
When you say
day.
no metabolism, you mean
Okay.
they are completely
You can
inert.
do it.
Completely.
Let's
I
work
mean, if you put a
today
virus
to
on
make
a table, it
tomorrow
will
a
sit
better
there
day.
forever.
For how long?
Okay.
Indefinitely. It doesn't
can
breathe,
do
it
it.
doesn't consume
Let's
nutrients,
work-
it doesn't generate heat, and it certainly doesn't create ATP for energy.
It has no power source.
No power source. It has no ribosomes, so it can't make its own proteins. It is, for all intents and purposes, a complex crystal.
The crystal.
It's like a USB drive. A USB drive isn't alive, right?
Yeah.
But it contains information that can crash your computer if you plug it in.
And in this analogy, the cell isn't just a house, it's the manufacturing plant.
That's a perfect way to put it.
The virus is a rogue blueprint that gets slipped onto the manager's desk.
Precisely. It's a blueprint that forces the factory to stop making cars and start making more blueprints. And this brings us to what the source material identifies as the golden rule of virology.
The golden rule.
If you want to understand why a virus causes a specific disease or why it spreads a certain way or how to stop it, you have to look at the equation. Genome type plus virion hardware, behavior.
Genome plus hardware equals behavior. That's our roadmap for the hour.
That's it.
So let's start with the first variable, the genome. The source material brings up the Baltimore classification. Now, to the uninitiated, this sounds like a police lineup.
It is a lineup of sorts. It was developed by David Baltimore, a Nobel laureate, and it was a brilliant insight. He realized that you shouldn't classify viruses by what they look like. Spiky, round, long.
The aesthetics.
Right, the morphology. You should classify them by how they solve one single universal problem, how they navigate the central dogma of biology.
The central dogma. That's the flow of information, right?
Yep.
DNA makes RNA, RNA makes protein.
That is the universal law of life on Earth. Your cells hold their master plan in DNA, keep it safe in the nucleus.
The vault.
They transcribe it into messenger RNA or mRNA when they need to build something. And then ribosomes read that mRNA to build proteins. Here is the catch. A virus must produce mRNA to survive.
Why mRNA specifically?
Because that's the only language the ribosomes understand. If a virus can't get its code into a format that your ribosomes can read, it's game over. It's just static. It can't build anything.
So mRNA is the bottleneck.
It is the universal bottleneck. Every single virus, no matter how weird its genome is, has to eventually funnel itself through the mRNA checkpoint. The Baltimore classification just categorizes them by the path they take to get there.
Okay, let's walk through these paths because this is where we see the logic you mentioned. The source material has this master integrated table. Let's start with class 1, double-stranded DNA or dsDNA. This includes the heavy hitters like herpes simplex, chickenpox, which is VZV, and Epstein-Barr virus.
Right. Class I is the most human-like in its logic. Our cells run on double-stranded DNA. That's our native operating system.
So it's a familiar file format.
It's a doc file in a Microsoft Word world. So when a herpes virus enters the cell, it's carrying a format that is natively compatible. It speaks the language fluently. So these viruses typically head straight for the nucleus.
And why the nucleus?
Because that's where the host keeps all the tools for reading and copying DNA. Our DNA polymerases are RNA polymerases. They don't need to pack a lot of exotic machinery because they just hijack ours. They walk into the library and use the photocopier that's already there.
That feels incredibly efficient.
It is.
Okay, what about class 2, single-stranded DNA, ssDNA, like parvovirus?
Similar logic, but a bit simpler. It enters as a single strand, but our nucleus isn't set up to read single-stranded DNA directly into mRNA.
There's a step missing.
A step is missing.
Yeah.
So it has to use host enzymes, our own DNA polymerases, to synthesize the second strand first, becoming double-stranded. Then it proceeds just like a class I virus. It's a small extra step to become legible.
Okay, now we leave the comfortable world of DNA and enter the RNA viruses. This is where things get alien.
This is where the cell's alarm bells start going off.
Class 3, double-stranded RNA.
Right. This is where the host defenses start to get suspicious. Human cells do not produce double-stranded RNA in their normal operations. It simply doesn't exist in us. So if a cell's sensors see double-stranded RNA,
It knows something is wrong. It's a red flag immediately.
it's a huge red flag. But beyond the detection risk, there's a mechanical problem, right? We talked about it. We don't have a machine to read it.
No photocopier for this format.
Exactly. We have enzymes to turn DNA into RNA. We do not have enzymes to turn RNA into more RNA. We can't copy it. So a virus like rotavirus, which is a major cause of gastroenteritis in children and is class 3, is holding a format we cannot process.
So what does it do? It's stuck.
It has to bring its own machine. It's a key principle of virology. If the host doesn't have it, the virus must bring it.
So it comes pre-packaged with its own tools.
It packs an enzyme called RDRPRNA-dependent RNA polymerase inside the viral capsid. It's like bringing a generator to a house that has no power outlets. It has to. It uses this enzyme to transcribe its double-stranded genome into individual strands of mRNA that the cell can finally understand.
So RDRP is the bridge. Without it, the virus is harmless.
Completely. If you could somehow inject just the naked genome of a class 3 virus into a cell without that enzyme, absolutely nothing would happen. It would just sit there until it was degraded.
That's a critical vulnerability then.
A huge one and a major target for antiviral drugs.
Let's move to class 4. Positive sense single-stranded RNA. The source lists polio, dengue, and famously SARS-CoV-2.
Right. Positive sense is the key term here. In virology, positive sense, or plus ssRNA, means the genetic code is already in the format of mRNA.
It's ready to go. It's the finished blueprint.
It is the blueprint. The moment a poliovirus or coronavirus releases its genome into the cytoplasm, the host ribosomes grab it and start building viral proteins. Immediately. They don't check the ID, they just see a valid code and execute it.
No conversion necessary.
None whatsoever. It is ruthlessly efficient. They don't need to do any enzymes with them because the genome itself is the instruction manual.
So class 4 hits the ground running. Now, compare that to class V, negative sense single-stranded RNA, minus ssRNA. There, it says influenza, rabies, measles. Negative sense always trips people up. What does that actually mean mechanically?
Think of it like a photograph and a film negative. Class 4, the positive sense, is the photograph. The ribosome looks at it, recognizes the image, and processes it.
Makes sense.
Class V is the film negative. It contains all the information, but it's inverted. If you feed a negative into a printer, you get garbage.
The cell can't read the negative.
No, it's gibberish to the ribosome. So class V viruses have the same problem as class 3. They are stuck unless they bring a developing studio.
Machine.
They must package the RDRP enzyme in the virion. They use it to transcribe that negative strand into a complementary positive strand. Only then does the infection truly begin.
That's a crucial distinction. Polio, a class 4, can travel light. Influenza, a class V, has to pack heavy.
That's the trade-off. It's a vulnerability, but it also allows for different replication strategies.
Okay, 2 classes left, and they're the really weird ones. Class 6, the retroviruses.
The hackers.
HIV is the classic example.
Right. Retroviruses are technically positive sense RNA, but they have a completely different ambition. They don't want to just use the factory.
They want to rewrite the company's charter.
They want to become part of the hard drive. They bring an enzyme called reverse transcriptase, or RT.
Which, as the name implies, goes against the central dogma.
It reverses the flow of information. It makes DNA from an RNA template. Then they use a second tool they bring, an enzyme called integrase, to physically cut the host's DNA and paste the new viral DNA code right into our genome.
And once it's pasted in,
it is there forever. It's a permanent modification. Every time that cell divides, it copies the virus along with its own genes. It becomes a permanent genetic resident, what we call a provirus.
that is just a fundamentally different level of invasion.
It's the ultimate form of persistence.
And finally, class 7, hepatitis B. This one seems like an oddball.
Class 7 is a strange hybrid, you're right. It has a DNA genome, but it's a weird, gapped, double-stranded circle. During its replication cycle, it makes an RNA intermediate and then reverse transcribes it back into DNA to package into new virions.
So it uses reverse transcription, but it starts with DNA.
Exactly. It's a roundabout way of doing things, but it works. The key takeaway is that it also relies on that reverse transcription process, similar to the retroviruses.
So that's the Baltimore classification. It's not just a list. It's a map of the logistics required to hack the cell.
It's a beautiful system for understanding the logic of infection.
But a genome is just software. The golden rule says we also need to look at the hardware, the physical tools of the trade.
Right. You can have the best code in the world, but if you can't get it inside the computer, it's useless. The hardware determines entry, exit, and even pathology. And the most important pieces of hardware are on the surface, the glycoproteins.
These are the keys, essentially,
Yeah.
or lockpicks.
They are the keys. A virus is locked out of the cell by the cell membrane. To get in, it needs to bind to a specific receptor on the cell surface. And this interaction is incredibly specific. It's like a key fitting into a lock.
And this specificity determines tropism.
Exactly. Tissue tropism. It's why you don't get the flu in your big toe. The receptors for influenza just aren't there.
Let's look at influenza again as the prime example here. We always hear about H1N1 or H3N2. Those letters and numbers, they stand for these proteins, right?
They do. The H is hemagglutinin. This is the entry key. It's a protein that's shaped perfectly to bind to a sugar called sialic acid, which coats the cells in your respiratory tract. Once hemagglutinin grabs that sialic acid, the virus is docked. It can initiate entry.
Okay, so that gets you in. What is the N?
The N is neuraminidase, and this solves a completely different problem that arises at the end of the infection.
The exit strategy.
Right. Imagine the virus has replicated. Millions of new virus particles are budding out of the dying cell. But the surface of that cell and the surfaces of the new viruses are still covered in that sticky sialic acid.
And the hemagglutinin on the new viruses is also sticky.
Exactly. The new viruses get stuck to the dying cell like Velcro. They clump together. If they can't break free, they can't spread to the next cell or to the next purser.
They're trapped.
So neuraminidase is a pair of scissors. Its sole job is to cleave the sialic acid, literally shave the cell bald of that sugar so the new viruses can float away.
So one protein is the grappling hook, and the other is the machete to cut the rope.
That is a perfect visualization. And this hardware dictates function principle applies to every virus. HIV uses a complex called GP120 and GP41 to bind to CD4 receptors on T cells.
The very cells that are supposed to fight it.
The irony is brutal. And herpes uses a fusion complex, GBGHGL, to bind to things like heparin sulfate. The hardware dictates the target.
The source material also mentions internal enzymes as part of the hardware, which we've touched on, the RDRP and reverse transcriptase.
The tools in the toolkit.
But there's a third category of hardware that I hadn't heard of before, viroporins.
Viroporins are fascinating and really, really dangerous. These are small viral proteins that assemble into a channel, a pore in cellular membrane.
They punch a hole.
A breach?
A breach that allows ions to flow freely. And ions are just charged particles, protons, which are H plus A, calcium, K2 plus I, potassium, K plus ion. Cells work incredibly hard to maintain very specific concentrations of these ions.
The ion gradient is life, basically. It powers everything.
Everything. Viroporins ruin that balance.
Why does the virus want to mess with the ions? What's the advantage?
In the early stages of infection, it helps with uncoding. So when a virus gets eaten by a cell, it's often trapped in a bubble called an endosome. The cell starts pumping acid protons into that bubble to digest what's inside.
The virus uses viroporins, like the M2 protein of influenza, to let those protons leak from the bubble into the virus particle itself. That acid bath inside the virus triggers a capsid to spring open and release the genome into the cytoplasm.
So it acidifies the room to break open its own box.
Yes. But later in the infection, these viroporins can accumulate in the host cell's main membrane, and this is where the neurovirology connection starts to creep in.
Because neurons are electrical.
They are fundamentally electrical. They run on precisely controlled movements of ions like sodium, potassium, and calcium. If you start punching holes in a neuron and letting calcium flood in or potassium leak out, what happens?
Chaos, you lose the gradient. The neuron can't fire properly.
Correct. You dysregulate the membrane potential. This can lead to neurons firing uncontrollably, which can manifest as seizures, or not firing at all, which can lead to coma. The source material highlights that this mechanism ion dysregulation from viroporins is a major cause of viral encephalopathy.
So it's not just the virus destroying the cell, it's a virus short-circuiting the electrical system of the brain.
It's an electrical storm, and that is a crucial insight. The hardware doesn't just help the virus replicate, it directly causes the pathology. It explains the symptoms.
Okay, so we've cased the joint, we understand the genome, And we've looked at the tools, the hardware. Now let's look at the break-in itself, the entry and replication logic. The outline lists 4 main ways they get inside.
We can view these as different heist tactics. The most common is probably receptor-mediated endocytosis.
The Trojan horse.
That's exactly what it is. The virus binds to the receptor, and the cell actively swallows it, thinking it might be a nutrient or a signal molecule. Influenza and dengue virus do this. The cell literally invites them in.
Unwittingly.
Unwittingly. Then there is direct membrane fusion. This is what enveloped viruses like HIV and herpes do. They're wrapped in a lipid skin they stole from a previous host cell.
A skull in disguise.
And that disguise is made of the same material as the target cell's membrane. So they just press up against the cell and their skin just melts into the cell's skin.
Like 2 drops of oil merging in water.
Yes. The capsid just falls gently into the cytoplasm. It's very stealthy, very elegant.
Then you have something less elegant. Genome penetration. The source says poliovirus uses this.
Right. Polio is a non-enveloped virus. It's just a naked protein shell, a capsid, no lipid skin.
So it can't fuse.
It can't
You can
fuse.
do
So it
it.
attaches to the surface,
Let's work
undergoes
today
a
to
massive
make tomorrow a
conformational
better
change,
day.
a shapeshift, that
Okay.
creates a pore
You
through the
can
cell
do it.
membrane. It
Let's
acts
work
like a
today to make
syringe,
tomorrow a
injecting
better
its RNA
day.
genome directly
Okay.
into the cell while
You
leaving
can
the
do
empty
it.
shell on the
Let's work.
outside.
That feels mechanical, brutal.
It is. It's a physical injection. And once they are in, regardless of the method, they have to decide where to set up the factory. We mentioned this briefly, but the logic is remarkably consistent.
DNA viruses go to the nucleus?
Generally, yes. They go to the nucleus because that's where the host's DNA replication and transcription machinery is. The exception is poxvirus, which is so big it brings its own entire factory and sets up in the cytoplasm.
And RNA viruses?
Generally stay in the cytoplasm. They bring their own RDRP, or their genome is the mRNA, so they have no need to go near the nucleus. They just get to work right away.
And retroviruses do a tour of both.
Right. They start in the cytoplasm to reverse transcribe their RNA to DNA. Then they have to traffic that DNA into the nucleus to integrate, and then the host transcribes it back to RNA, which goes out to the cytoplasm to make proteins. They use the whole cell.
Okay. We understand the machine. We understand the break-in. Now I want to move to the section of the source material that feels the most profound and, frankly, the most unnerving. Section 4. Neural latency. The hideout.
This is where we move from acute virology to chronic neurovirology. Most infections we get, like the common cold or the flu, are acute.
Yeah.
The virus comes in, replicates like crazy, the immune system gears up, kills it, and you recover.
Hit and run.
A hit and run. But some viruses play a longer game. They find a sanctuary where the police, the immune system, can't easily go.
And that sanctuary is the nervous system.
Right.
Why neurons? Of all the cells in the body, why are they the perfect hideout?
It comes down to the unique biology of the neuron. First, and maybe most importantly, neurons are postmitotic.
Meaning they don't divide.
They don't divide. If a virus hides in a skin cell, that cell will eventually die and slough off, or it'll divide and the viral genome might get diluted or lost. But a neuron, you have the same neurons today that you had when you were 5 years old.
It's a hideout that lasts a lifetime.
Exactly. If you get your genome into a neuron, you have tenure. You are set for life.
So that's the longevity. What's the other piece?
The other piece is immune privilege. Specifically, neurons have low MHC-I expression.
Okay, let's break that down. MHC class I is the window, right? The display case.
It is the cell's personal display window. In a normal cell, say a liver cell or a lung cell, these MHC-I molecules are constantly taking little bits of all the proteins being made inside the cell and putting them on the surface for the T cells to inspect.
The immune system's police patrol.
Right. And if a T cell, a cytotoxic T lymphocyte, sees a viral protein in that window, its orders are simple. Kill the cell. Terminate it to prevent further spread.
A simple, brutal inspection.
But the brain is different. The central nervous system is, it's precious. You cannot afford to be constantly killing neurons. They don't grow back. So evolution has made a trade-off. Neurons deliberately downregulate MHC-I. They keep the curtains drawn.
So the T cells walk by, they know a neuron is there, but they can't see what's happening inside.
Exactly. They can't verify the infection, so they don't attack. This allows viruses like herpes simplex and varicella zoster to sit inside the ganglia for decades without being destroyed. It's an immune blind spot.
The source material draws a very sharp distinction here between latency and persistence.
Yeah.
I feel like we use those words interchangeably in everyday life, but mechanically they are different.
Very, very different, and it's a critical distinction. Latency is total silence. The viral genome is sitting in the nucleus, often as a circular loop of DNA called an episome. but it is not making proteins. It's transcriptionally silent.
So there's nothing to show in the window.
There's nothing to put in the window. Even if the curtains were open, the virus is a ghost. It's just a piece of silent DNA.
So it's truly invisible. What's persistence then?
Persistence, on the other hand, involves a low-level smoldering infection. The virus is replicating very slowly, or it's producing just a few non-essential proteins. This attracts a chronic, low-level immune response. This is what we see in conditions like SSPE, subacute sclerosing panencephalitis, which is a rare but fatal consequence of a measles infection.
So it's not silent.
It's not silent. It's a quiet riot that causes progressive damage over years. Latency is a sleeping agent. Persistence is a years-long siege.
Let's look at the most wanted list of these neurotropic viruses. Where exactly are they hiding? The source lists specific anatomical locations, which is fascinating.
Yes, the geography matters immensely. Take HSV-1, herpes simplex 1. This is the virus that causes cold sores.
Right.
After the initial infection, usually on your lip, the virus retreats. It travels up the sensory nerve and goes to sleep in its reservoir, the trigeminal ganglia. It's a big cluster of nerve cell bodies right near your temple, deep in the face. It's the hub for facial sensation. The virus just sits there, latent. Then when you get stressed or get a sunburn or have a fever, some trigger causes it to reactivate. It wakes up, travels back down the exact same nerve axon, and causes a blister on the lip.
So the blister is just the tip of the iceberg. The base of operations is deep inside my skull.
Correct. The skin lesion is just the endpoint of a nerve highway. And HSV-2, genital herpes, uses the exact same logic but hides in the sacral ganglia at the base of the spine, which serves the genital region.
Same strategy, different geography. What about chickenpox?
VZV.
Varicella zoster virus. We all get chickenpox as kids, hopefully. The spots heal, but the virus doesn't leave. It travels from the skin lesions to the dorsal root ganglia.
And nerve clusters along the spinal cord.
Exactly. The ones that control sensation for your entire torso and limbs. It sleeps there for 50, 60, 70 years. Then as your T-cell immunity weakens with age, it wakes up.
And because it's in a specific nerve root, a specific ganglion, it erupts along the specific strip of skin that nerve serves. It's called a dermatome. That's why shingles forms that characteristic painful band. It's literally tracing the wiring diagram of your nervous system on your skin.
That is wildly creepy. Now, HIV is different. It's neurotropic, but it doesn't hide in the neurons themselves, right?
Correct. HIV infects immune cells. In the brain, its main reservoirs are the microglia and macrophages. These are the resident immune guardians of the brain. The virus integrates its DNA into their genome as a provirus.
So it hides inside the guards.
It hides inside the guards. This leads to what's called hand HIV-associated neurocognitive disorder. And it's a huge problem because even if antiviral drugs clear the virus from the blood, the brain remains a sanctuary site. Many of those drugs can't cross the blood-brain barrier effectively to get to those infected microglia.
And then there is the absolutely horror story, rabies.
Rabies is unique. It doesn't really do latency in the same way. It just moves very, very slowly.
It's a slow burn.
It enters a nerve ending in your muscle, say from a dog bite on your hand, and it hijacks the cell's own internal transport system. It latches onto molecular motors, dynein motors, and gets a ride up the nerve axon.
Slow axonal transport.
It's a slow, inexorable march. It can take weeks or months, but once it reaches the brainstem and begins replicating, it is almost 100% fatal. It's the most deadly pathogen known to man once symptoms begin.
There is an aha moment in the outline here regarding the damage these viruses cause. I asked the question, if the virus is just waking up, like in shingles, why does it hurt so much? Is the virus actually eating the nerve?
You would think so, wouldn't you, that the virus is just chewing up the neuron. But no. The core principle of neurovirology, especially with reactivation, is that the damage is primarily immune-mediated.
It's friendly fire.
It is almost entirely friendly fire. When the virus wakes up and starts making proteins again, the immune system finally sees it. It panics. It floods the nerve ganglion with inflammation, T cells, and cytokines to contain the breach. It is that intense immune response in a very sensitive, confined space that damages the nerve, causing the intense pain of shingles or the facial paralysis of Bell's palsy, which can be caused by herpes reactivation.
That explains why the treatment is sometimes steroids, corticosteroids. They turn down the immune system.
Exactly. You were trying to stop the SWAT team from burning down the house to catch the burglar. You're accepting a little more viral replication in exchange for less neural damage.
Okay, let's transition to that SWAT team, Section 5. The host immune response. How does the body fight back when it does detect an intruder? The source describes a stepwise cascade.
It's a highly choreographed military operation. Step one is detection. We talked about how the cell knows something is wrong. Inside our cells, we have tripwires, proteins like RIG-I and a family called toll-like receptors, or TLRs. They are pattern recognition scanners.
What are they scanning for?
Alien formats. They're hardwired to look for things that shouldn't be in a human cell. They look for double-stranded RNA. They look for DNA floating in the cytoplasm where it shouldn't be. If RIG-I grabs onto a piece of viral dsRNA, it snaps shut like a mousetrap and pulls the fire alarm.
That leads to step two, the alarm, the interferons.
Right. The infected cell starts screaming. It releases signaling molecules called type I interferons, specifically interferon alpha and interferon beta. This is a neighborhood watch signal. The infected cell is basically yelling, "I'm compromised, lock your doors."
And what does the neighbors do? They get the signal.
They get the signal. The interferon molecules float to the neighboring cells and bind to their receptors. This triggers an antiviral state. They literally stop making most proteins, they turn on enzymes that chew up RNA, they lock their doors, they sacrifice their own productivity to prevent the virus from finding a new home in them. It creates a firewall around the initial infection.
Then comes step three, the fire, the cytokines.
If interferon is the alarm, cytokines are the call for reinforcements. Molecules like IL-1, IL-6, TNF-alpha, these are the chemicals that cause inflammation. They make blood vessels leaky so immune cells can get into the tissue.
And they also cause fever.
They do. They travel to the hypothalamus in the brain and turn up the body's thermostat. A fever is a weapon. It slows down viral replication and speeds up the metabolism of our immune cells. But here is the neural connection again. These cytokines directly modulate neural circuits. They cause what we call sickness behavior.
The brain fog, the fatigue, the overwhelming desire to just curl up in a ball and do nothing.
That isn't a side effect, it's a centrally mediated strategy. The cytokines are reprogramming your brain to force you to conserve energy so the body can dedicate all its resources to fighting the infection. You aren't just feeling sick, you are being chemically restrained for your own good.
Wow. And finally, step four, the soldiers. The actual cells arrive.
The cellular response.
Yeah.
NK cells, natural killers, often show up first. They're the early responders. They kill anything that looks stressed or has suspiciously low levels of MHC-I.
The cells with the curtains drawn.
Exactly. Then the CD8 cytotoxic T cells arrive. They are the snipers. They meticulously check the MHC windows and execute infected cells with precision. And all the while, CD4 helper T cells are coordinating the entire battle, and B cells are getting the signal to start pumping out antibodies to gum up the viral surface proteins so they can't infect new cells.
It's an amazing multi-layered system. But sometimes it's not enough or it's too slow. We need reinforcements. Let's talk about Section 6, medical countermeasures, antivirals, and vaccines.
Right. And antivirals are incredibly difficult to design. Remember, the golden rule viruses use our hardware?
So if you poison the machine, you poison the patient.
Exactly. You can't just throw sand in the gears because they are our gears. So effective antivirals must be exquisitely specific, targeting the tiny percentage of the process that is uniquely viral.
The targets we identified in the hardware section. The viral-specific enzymes.
Yes. Take acyclovir, the classic herpes drug. It targets the viral DNA polymerase. It's a nucleoside analog. It looks like a building block of DNA, specifically guanosine, but it's a dud. It's missing the piece that allows the next block to connect.
It's a fake brick in the wall?
It's a chain terminator. When the viral enzyme is rapidly building a new viral genome and tries to use acyclovir, the wall stops growing, the chain is terminated, and crucially, our human DNA polymerase is much smarter. It can spot the fake and ignores it, but the viral one can't. That specificity is what makes the drug safe.
What about protein inhibitors? We hear about those with HIV.
So we talked about how some viruses, like HIV in class 6, make one giant long protein chain, a polyprotein that needs to be cut up into its functional parts. The viral protease is the molecular scissors that does the cutting.
Right.
Drugs like ritonavir are designed to perfectly fit into the active site of those scissors and gum them up. So the virus produces its long protein chain, but it can't cut it into the functional parts. The new virus particles remain immature and non-infectious.
And then there are the drugs for influenza, like Tamiflu.
Oh, siltamivir, yes. It inhibits neuraminidase. Remember the machete that cuts the virus loose from the cell surface? Tamiflu dulls the blade. The new viruses get made, but they remain tethered to the dying cell. They clump together, can't spread, and the immune system cleans up the mess.
But there is a major limitation noted in the source material, the blood-brain barrier challenge.
This is a huge, huge issue for neurovirology. The brain is wrapped in a very tight layer of cells that acts as a bouncer, filtering the blood. Many antiviral drugs are large, complex molecules. They get stopped at the door. They just bounce off the barrier.
So the virus can be hiding in the brain, and the drug simply can't reach it.
Yes. This is a massive problem with HIV. You can suppress the virus in the blood to undetectable levels, but it can still be smoldering away in the microglia in the brain because the concentration of the drug is too low in the central nervous system. It remains a sanctuary site.
And the other major limitation you mentioned, antivirals don't cure latent infections.
They cannot touch them. If the virus is in that silent episome state, just a quiet loop of DNA in the nucleus of a neuron, there is no enzyme working. There is no polymerase to jam, no protease to block. The drug has no target. This is why you have herpes for life. We can treat the outbreak when it's active, but we can't go into the library and burn the book.
Which brings us to vaccines. If we can't cure it, we have to prevent it in the first place.
Pre-programming is the best way to describe it. A vaccine is a wanted poster that you show to the immune system. You show it the face of the enemy, the antigen.
You can do
so
it.
it builds
Let's
an army
work
of
today to
neutralizing
make tomorrow
antibodies
a better
and memory
day.
T cells
Okay.
before the real infection
You
having
can
any
do
bits.
it.
The source outlines
Let's
the menu
work
of
today
vaccine
to
types.
make tomorrow
Live,
a better
attenuated,
day.
inactivated,
Okay.
subunit, and
You
now
can
mRNA.
do it.
It's a trade-off
Let's work.
between safety and power. Live attenuated, like the MMR vaccine, is a crippled, weakened virus. It's alive, it replicates a little, so it creates a massive, roaring immune response. It's the best teacher. But you can't give it to immunocompromised people because even the crippled virus might still win.
Been inactivated, like one of the polio shots.
The Salk vaccine, yes, the virus is dead, chemically killed, it's very safe, but the immune system doesn't panic as much, so the memory can fade. You often need boosters.
And the new era, mRNA vaccines.
This changes the entire logistics. Instead of spending months growing viruses in chicken eggs like for the flu shot, you just synthesize the code for the viral spike protein. You put that code in a little lipid bubble and inject it. Your own cells read the code, build the spike protein, display it in the MHC window, and train the immune system from the inside out. It's incredibly fast and adaptable.
The source material has a specific neuroinsight regarding vaccines that I thought was important. They reduce neuroinvasion.
This is absolutely vital. By having high levels of neutralizing antibodies already floating in your blood from a vaccine, you catch the virus before it finds a nerve ending. You stop the viremia, the presence of virus in the blood. Once a virus like rabies or polio enters a nerve, antibodies can't really touch it anymore. The vaccine builds a wall around the nervous system.
But the enemy adapts. We have to talk about the mutation problem.
Evolution in real time. RNA viruses, especially those in class IV that use RDRP to copy themselves, are notoriously sloppy.
Their photocopier is bad.
Their photocopier is terrible. It lacks a proofreading function. It makes typos constantly.
Most of those typos must be harmful, right? They break the code.
Most are lethal to the virus, yes. But every once in a while, a typo changes the shape of a surface protein, like hemagglutinin, just enough that the antibodies you made last year don't stick to it anymore. That's antigenic drift. That's why we need a new flu shot every year. The target has moved slightly.
And then there's the big one, antigenic shift.
That is the catastrophic event. Influenza is special because it has a segmented genome. Its genes are on 8 separate pieces of RNA, like a deck of cards.
Okay.
If a single cell, say in a pig, gets infected with a bird flu virus and a human flu virus at the same time, the viruses can shuffle their decks as they replicate.
You get a hybrid.
You get a Frankenstein virus. A completely new subtype with a new combination of H and N proteins that no human immune system has ever seen before. This is what causes pandemics like in 1918 or 2009.
We are reaching the end of our journey through the source material, but we have to talk about the final section, evolution. The relationship between hosts and viruses isn't just a war, it's a history.
It really is. We tend to think of it as us versus them. But the source points out a truly mind-blowing fact. 8% of the human genome is derived from endogenous retroviruses.
8%. That is a massive number. That's significantly more than the percentage of our DNA that actually codes for our own proteins.
Think about what that means. These are the fossil records of ancient viral infections from millions of years ago that infected the germline, the egg, or sperm of our distant ancestors. They integrated their DNA, but then they broke. They mutated and got stuck. We have been carrying their corpses in our DNA for eons.
But we didn't just carry them as junk DNA. We repurposed them.
We did. We domesticated some of their genes. The most famous example is a gene called syncytin.
Syncytin. What does it do?
It is absolutely essential for the formation of the human placenta. It's a protein that causes cells to fuse together to create the syncytia trophoblast, the critical barrier between the mother and the fetus that allows nutrient exchange but blocks immune rejection.
Fusion. That sounds very familiar. That's a viral function.
It should. Syncytin is originally a viral envelope fusion protein. We stole the fusion tool from an ancient retrovirus. The virus used it to melt cell walls to infect us. We now use it to fuse cells to build a safe home for our developing offspring.
So without that ancient viral gene,
Mammals as we know them likely wouldn't exist. You, me, all of us. We literally would not have been born without viral DNA being part of our own genome.
that recontextualizes everything we've been talking about. We aren't just fighting them. We are, in part, them.
In a very real way, yes. The viral operating system didn't just hack us. Over evolutionary time, we hacked it back. We assimilated it. And that, to me, is the ultimate lesson here. Viruses aren't just destroyers. They are sources of profound genetic innovation. They are the open source code that evolution borrows from to build new things.
So we've covered the hardware of the capsid and the glycoproteins, the logic of the Baltimore classes, the hideout of latency in our nerve ganglia, and now the legacy written into our own genome.
Yeah.
It really does challenge the simple definition of life.
It does. A virus is just a geometric cage with a code inside. It's dead matter. Yet it drives human evolution, it shapes our immune system, and has found a way to persist in the most protected sanctuary we have, our own nervous system. The battle isn't just external, it is intimate.
The battle is intimate. I love that. And it's a reminder that the next time you get a cold sore or hear about someone getting shingles, you're not just seeing a rash. You're seeing a sleeper agent waking up from a decades-long nap inside the architecture of your nerves, following a logic that is older than humanity itself.
Precisely. Stay curious.
Thank you for guiding us through this. This has been The Deep Dive. We'll see you next time.